Hi, thanks for joining us today. My name is Ronnie Powers. I'm the founder and CEO of Pluto. Today we'll be discussing differential expression analysis in Pluto. Differential expression analysis is a common kind of comparison that you might want to do to look at genome-wide gene expression profiles in two groups of samples, particularly in experiments like RNA sequencing. So we'll be looking at a bulk RNA-seq example today in Pluto. And to set you up in this experiment, you can see in my sample data table here, the types of samples that we have in this study involve samples that were collected from either healthy controls of varying ages or CoV-2 positive cases. We also have annotations for sex. We know that some of the patients were male, some were female. So we have that assortment in this study. To run differential expression analysis in Pluto, now that this data has been uploaded, you'll see I just clicked this Add Analysis button to get started. I'll select Differential Expression Analysis and Next, and here we're going to be setting up our analysis parameters. Particularly for differential expression, what we care about are the groupings of samples. So in this case, you'll see I have different biological factors that could be used to group samples. I can group them by condition or by sex. In Pluto, the variables in this analysis pane here are data-driven based on the sample data that you uploaded. So I could, for example, group my samples just by condition. In this case, I have the option of selecting our CoV-2 positive patients as my experimental group, and then the healthy controls as the control group. I can click Run Analysis to kick off a differential expression analysis pipeline. You'll see by default that pipeline produces data that we're showing right now on an interactive volcano plot. So I can see the genes that have been differentially expressed. Genes on the right side of this volcano plot increased in expression consistently in the CoV-2 group, and genes on the left side of this plot decreased in expression consistently in the group of CoV-2 patients. Now, we've built Pluto to be very flexible to the different kinds of comparisons that you might be interested in running. So, because we knew that sex was a biological variable in this clinical study that I've got, we can clone this analysis and perform an even tighter comparison. So now that I've cloned that, on my active copied plot here, I'm going to edit the analysis so that we're not just grouping by condition, but also grouping by sex. You'll see that the groups were recalculated, so now I can choose CoV-2 female samples in my experimental group, and then they're matching control females as the control group. Now when we click Run Analysis to update this comparison, you'll see the volcano plot changes shape a little bit. What we're looking at now are increased genes in the CoV-2 female as compared to control female samples only. In both cases, these plots can be customized using the Plot tab. For differential expression data, we can visualize the data as a volcano plot like we're looking at now, or as a heat map. We can control the plot title or even change the statistical significance that we're using and show that with an adjusted p-value threshold line. So I can make our threshold much lower in this case and show the p-value threshold line. Come down here to click View Changes to update the plot. Now you'll see a threshold for my p-value on the y-axis, and then the plot, the points that are no longer meeting that threshold are not colored. I might be interested in this case in looking at genes of interest that are consistently changed in both the male and female, or in this broad comparison and the tighter comparison. So I can choose to label certain targets, for example, CXCL10 and 11 on my volcano plot, and again, click View Changes to see those targets highlighted. And finally, I can customize the appearance of this plot by editing the colors for the different groups. In this case, on a volcano plot, we have increased and decreased genes, and you'll see I can select any color for those genes from the hue picker, from my own favorite colors, which I can manage in Pluto, or if your organization has a subscription to Pluto, in organization-branded color palettes, which are managed here. So I'll select some custom colors for these increased and decreased genes now on my volcano plot and click View Changes to update. So now we're showing two different volcano plots that we've run for differential expression analysis. The underlying results and data that are shown on these plots can be searched and downloaded from the Results tab. Because we have two differential expression analyses that were run, you'll notice that I can switch between them and view either table here from the Results tab. I can search and even read a little bit about the biological annotations here for these different genes. These DEG tables can be downloaded, of course, in a tabular format for your own analysis. For each analysis in Pluto, in addition to the results table, you can view the methods. These dynamic methods sections will update to reflect any of your analysis parameters, for example, the exact comparison that I ran, or that p-value threshold that you saw me modify. Easily copy this methods section to paste it into a paper or a presentation that you're working on. This was your brief overview of differential expression analysis in Pluto. Thanks for watching, and we'll see you in the next one.